This is episode 250 of the Stem Cell Podcast, Blastoid Development and Implantation with Dr. Nicolas Rivron. Hey, everybody, we are Daylon and Arun back with another episode of the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Stem Cell Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, as our guest, we have Dr. Nicolas Rivron from the Institute of Molecular Biotechnology in Austria. He's on the show today to talk about his groundbreaking research on blastoids and self-organization. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, we'd like to remind our listeners about ESC and iPSC News, one of STEMCELL's free weekly scientific newsletters. ESC and iPSC News summarizes all of the latest research, news, jobs, and events in ESC and iPSC research and delivers it right to your inbox every Wednesday. Save time and keep current with ESC and iPSC News, and you can subscribe for free at www.escellnews.com.
Talking about groundbreaking, Arun, I'm leading off the roundup today with a truly groundbreaking watershed moment in our field. This is, of course, the results that were recently announced by Blue Rock from their phase 1 study. Results were presented at the International Congress of Parkinson's Disease and Movement Disorders that was in Copenhagen, Denmark, here at the end of August. And, you know, this is, everyone's been waiting for these results since the beginning, really. The culmination of decades of work, and it's a story about bemdaneprocel. I mean, first we got to talk to Blue Rock about, or anybody, about how they formulate the names here. I'm going to struggle to say that a few times during this roundup story, so bear with me. But bemdaneprocel, it's this investigational cell therapy that's meant to replace dopaminergic neurons that are lost in Parkinson's disease, and it's made up of these dopaminergic neuron precursors derived from human embryonic stem cells, and then they are surgically implanted into the brain where they can reform neural networks and restore motor and non-motor function to patients. And, you know, there's a huge need here. Estimated more than 10 million people worldwide suffering from Parkinson's disease, about 1 million of those living in the United States alone. And we know there's no cure that's effective, and the treatments that are out there, they kind of lose efficacy over time. So this phase 1 study was a multi-center, multi-site, open-label, non-randomized, and non-controlled study that was focused on an initial cohort of 12 patients that were divided into 2 different dose levels. The first level was cohort A, 5 subjects that received a dose of 0.9 million cells for caudate putamen, which is where the cells are injected. The second cohort was 7 subjects, reaching a total of 12, and those received triple the dose, 2.7 million cells per caudate putamen. And the assessments of these patients were meant to progress over the course of 2 years. This is 1 year out, where we're looking at the primary objective, demonstrating safety and tolerability. And that objective was met in all 12 participants, both the low and high dose, with no serious adverse events. There were 2 adverse events that were reported that were unrelated to the cells. One was a seizure that was attributed to the surgical procedure. One was a patient got COVID. I mean, can't blame anybody for that. Both of those resolved without any sequelae. PET imaging, and this is big, showed that there was evidence of cell survival and engraftment in both the low and high dose cohorts at a year out. And efficacy, functionally, this is the most exciting to me, using this thing called the Hauser diary, which categorizes patients as being in the on state when their symptoms are controlled, the off state when they're experiencing a worsening of their symptoms. So in the high dose cohort, the patients reported an average improvement of 2.16 hours in the on state, and that corresponded with 1.91 hours in the off state. So that made sense. And here's the key for me. In the low dose, there was also an improvement, but it was about a third of, almost exactly a third of the efficacy, 0.72 hours reported in the on state. That is without dyskinesia, and corresponding decrease of 0.75 hours in the off state. So intuitively lines up for me. You're getting a third the dose of cells, you're getting a third the efficacy. I know I'm oversimplifying there, but there was also this objective measure, or more objective measure, from the Movement Disorder Society called the Unified Parkinson's Disease Rating Scale that showed similar reduction of symptoms in these patients, and scaling similarly better in the high dose cohort, and improvement, but not as great in the low dose cohort. So for me, this is a home run. All you can expect from a phase 1 study. I mean, notwithstanding they walk out of the clinic and never have dyskinesia again, but that's pretty far-fetched. I think showing any improvement at this point is a major milestone. And of course, everybody, I think, is really excited about Blue Rock, planning already underway for phase 2 of the study that's going to be enrolling patients in the first half of 2024. So right around the corner. And, I can't embellish these results enough. This is such a watershed moment in our field, as I mentioned before, and it's so great to have it be positive. The fact that we actually have some efficacy results here, not just safety and tolerability, I think is really going to engender a lot of enthusiasm and keep momentum going for other clinical trials as well as this one. Arun, I've been waiting for this moment forever. What are you thinking?
Yeah, this you said it all. This is really just a watershed moment in the field. I mean, so many years of incredible basic science work leading to the development of this particular therapy, from originally from Lorenz's lab, and then the work at Blue Rock. Really incredible to see no major safety issues at all in these 12 participants in this low dose. And I think the other thing, and again, this isn't necessarily my area of expertise, but like you alluded to, seeing some dosing related responses in these patients is also really promising, because maybe in the future, even if you increase the dose even further, then you'll get an even better amelioration of the disease phenotype here. So that's really cool to see. And I mean, it's one thing to announce the results here. I really want to see the inevitable publication that comes out of some of this initial data, and I'm sure it's going to be coming very soon. And of course, looking to the future and thinking about that phase 2 study and seeing how that's going to go. I mean, you just want to keep seeing this thing scale up and up and up, and seeing the safety and the efficacy ultimately improve and be consistent with the scaling of the trial. So can't wait to see where this goes.
Yeah, I agree with you there. I can't wait to see with more granularity the distribution of the efficacy results across patients. You're injecting something into the brain cells. It's a live product. You can't expect it to behave the same way in each individual case. So I'm really interested to see the distribution there. And by the way, like, we're talking about a neurodegenerative disease. So the fact that these patients are improving is a big deal. They're not just not getting worse, are static, they're getting better. And this is a trial where they're going to continue to monitor these patients for another year after this. So I hope to see, even, I guess, consistent, maybe improvement beyond the efficacy already, but at least a static result. These patients aren't going to get worse.
Moving on to another very clinically relevant story. This is actually coming from the New England Journal of Medicine. I alluded to in the previous story, where I like to see the clinical trials being published, the actual publications and the raw data of the clinical trials in journals, just to validate that what we're saying in these press releases actually holds up in a publication. And here is a New England Journal article from a Novartis associated study using CRISPR-Cas9 genome editing of the HB1 and HB2 promoters to actually treat sickle cell disease. And certainly treating sickle cell disease using gene therapy and gene editing approaches, it's not a brand new concept. This has been done in various capacities over the last few years, initially using like lentiviral based approaches for correcting CD34 positive hematopoietic stem cells ex vivo and then retransplanting them back into the patients. This is really cutting edge because, of course, you're incorporating CRISPR into the equation and perhaps having a more specific targeted editing approach for restoring and ameliorating some of the clinical phenotypes associated with sickle cell, which is, of course, such a painful and devastating disease, predominantly affecting African Americans, but also other populations as well. So we know that the backstory of sickle cell by now, right? It's caused by this defect in the beta globin subunit of adult hemoglobin. You have this sickling condition creating deformed red cells that hemolyze and cause all sorts of downstream symptoms and really painful phenotypes, which lead to progressive organ damage and ultimately early death, right? And the thought is you can elevate fetal hemoglobin levels in red cells to actually protect against some of the complications of sickle cell disease. So there's this technology, this cell product, OTQ-923, again, developed in conjunction with Novartis, which is using CRISPR-edited CD34 positive hematopoietic stem and progenitor cells, which specifically have been, the CRISPR is targeting the HBG1 and HB2 gamma globin gene promoters. And this is the fetal version of the globin subunit of hemoglobin. So basically the thought is you can target the upstream promoters of the HBG1 and HBG2 to basically knock out a regulatory region which ultimately reactivates the fetal globin gene, right? And that's their approach to alleviate some of the clinical manifestations of the disease. So turn off the adult problematic version of hemoglobin and turn on the fetal gamma globin subunit, okay? And so they talked about how they did the upstream to actually identify the best guide RNA that can be used to specifically target those promoter regions upstream of HBG1 and HBG2, these gamma globin gene promoters, right? So they found this guide RNA 68 after doing this huge guide RNA screen, right? Which is great. They did a bunch of preclinical validation that that's the best guide RNA for their targeting approach. And then, the crux of the paper, and again, the major reason why this is a New England Journal article, and we don't cover too many of these New England Journal approaches or publications, right? You got to put these things into patients. That's the whole point of publishing something in New England Journal is the results of a clinical trial, right? So they enrolled patients, participants with severe sickle cell disease in a multi-center phase 1 clinical trial study, analogous to what the Blue Rock folks are doing. Very early phase, safety and efficacy, safety mostly, looking at the safety of this OTQ-923 CRISPR-edited CD34 positive cell product, right? So what do they find? They found that one, like I talked about, the guide RNA 68, they validated that was the best guide RNA for their targeting approach. And in the study, there's only 3 participants here that they're talking about. Only 3 participants who actually received the autologous OTQ-923 after their mild ablative conditioning. And they followed these individuals who had severe sickle cell for 6 to 18 months. And importantly, at the end of the follow-up period, all the participants had the engraftment and the stable induction of the fetal hemoglobin caused by, again, the CRISPR-based modification. There was an increase in the fetal hemoglobin as a percentage of the total hemoglobin, going up, 19 to 27%. The fetal hemoglobin broadly was distributed in the red blood cells, and ultimately, and the most important thing here is the manifestations of sickle cell disease were decreased during the follow-up period. So that's the most important thing for the patients, that of course they feel better, their disease phenotypes are alleviated, and that on the basic science side of it, this is a really good study reflecting the power again of CRISPR in vivo modification, modifying these CD34 positive hematopoietic stem and progenitor cells do what ultimately they're supposed to do, which is to alleviate the symptoms of this severe sickle cell disease. Really kind of an ingenious based CRISPR modification approach of these gene promoters. All you got to do, you didn't have to do any sort of correction here. This was just using a guide RNA, as like I've said before, the analogy is taking a shotgun to the genome and just knocking out the portion of the HPG1, HPG2 gene promoters that are normally involved in the silencing of those two genes. So I think because it's not an HDR or homology directed repair approach, it's much easier to do. Anytime you involve correction of a genomic region, these approaches get a lot more complicated. So I think low-hanging fruit in some way, right? But ultimately, the most important thing is showing the alleviation of the clinical phenotypes for this really devastating disease in sickle cell.
Yeah, man, whatever works, right? And I love hematopoiesis. We all know that, but this idea, I think, is why I fell in love with not just hematopoiesis, but hematopoiesis as a means of therapy. When Stu Orkin was kicking around this idea early days, it was about when I was coming into the field. And I remember falling in love with just that idea of the hack. It made me really appreciate an idea that has recurred over and over in my career, and just is generally I think a very closely and a treasured element of science and innovation, which is hacking the system, right? It can't fix the adult globin, reactivate the fetal globin. I thought that was such a beautiful idea, and since then there's been a lot of variable ways to achieve that goal. A lot of ways to skin that cat. And not even to mention the more classic genetic therapies for treating sickle cell. So as you said, this is kind of low-hanging fruit, less complex, I think more efficient, I guess. My question now is, with all these different ways of skinning the cat, all these different hacks, which is going to rise to the top? There's got to be a point at which one of these therapies becomes the dominant modality, just from, like, I guess a economic commercial standpoint. And back to your point, I wonder if it's going to be this fetal globin activation as opposed to like a homology directed repair of the sickle cell characteristic, because it's got to be cheap. And as we've talked about also on the show, you got to get in there early. You don't want someone living with the sequelae of sickle cell their whole life, and the inflammation that goes with it. It may degrade the hematopoietic system. So anyone with sickle cell with a cheap therapy could get in there early and have a life where they're spared disease and associated sequelae. Yeah, whatever works. I'll take this. It looks like it's very promising.
Yeah, definitely very promising. And I think one of the perhaps negatives, but of course you have to do these studies the right way, right? These trials take time, right? So developing any sort of cell therapy, genetic modification, gene therapy, whatever, you have to go through the rigors of clinical trials. And this publication is likely a decade in the works, right? And certainly there are newer technologies in addition to CRISPR-Cas9, which can, perhaps modify these cell products even more specifically than CRISPR can. We know that's possible. CRISPR, in a lot of ways, CRISPR-Cas9 is old school now, which is amazing to think about, because it's just that technology has only been around, at least in the everyday use, for about 10 years now. But now we're talking about adenine base editors, the prime editing approaches, which inevitably are going to also be used for some of these correction approaches. It's in the pipeline. It just takes time, right? Perhaps because this approach has shown so much promise, those downstream approaches are going to be accelerated. I don't know, to some extent, right? Ultimately, we have to see what's going to win out, or if this is really going to be iterative. There was the Lenti approach, and that worked, so that became mainstream. But then CRISPR came out, that became mainstream, and that's the gold standard. Whatever, who knows what's going to happen 10 years from now? You can't hope to do all of these trials at exactly the same time, because of course the technology is emerging at different times. But we'll see. That's I think regardless, just having a lot of arrows in the quiver, so to speak, for treating sickle cell is a good thing.
Yeah, CRISPR, dated. need to come up with something new. But along those lines, I got a segue there. I got the next thing here, which is adenine base editing using CRISPR. Maybe a little bit more current for your taste there, Arun. But generally, what we're talking about here, and what we've been talking about and are going to be talking about for the foreseeable future, is precision medicine, personalized medicine, right? And in this case, not just treating the patient and their specific disease, specific mutation in the case of sickle cell. But I got a story about treating a patient's specific cancer, right? Not just the patient, but their specific cancer. And that's CAR T. There's all these targeted immunotherapies based on a lot of different immunomodulations, but CAR T, I think, is above all the most romantic to me. The idea of this, soldier you send out to go hunt down the tumor cells. I love it. And it's been, used widely, clinical trials for solid tumors now too, but it's revolutionized the treatment of hematologic malignancies, pretty much overnight, changing the way we treat. Not overnight, as Arun alluded to, these things take years, but has truly revolutionized the treatment of hematologic malignancy with high rates of cures for cancers that have typically been resistant to traditional chemo. But there is some challenges there, right? When you're treating the tumor cells with CAR T, these hematologic malignancies, they got to be specific to the cells, right? So if it's B-cell leukemia, lymphoma, you have CD19. If it's myeloma, you have BCMA. If it's T-cell leukemia or lymphoma, you have CD7. For AML, it's CD33, right? And that's been working. But there's some challenges technically, but also from like a economic kind of drug development standpoint, it's not very efficient, right? And you're also limited in terms of antigens, because you got to have antigens as a room. We always talk about the off-target stuff, right? You got to have antigens that are going to be present and specific to the tumor, but not going to be in other vital places where the CAR T will wipe you out, the good cells, right? So that's a challenge. You got to have something that's clinically tolerable. And that's not always going to be the case for every tumor. So as an alternative, at least with hematologic malignancy, there's this idea floating around of attacking just a pan antigen, in this case, CD45. It's expressed on the surface of pretty much all hematopoietic cells, right? But of course, because it's expressed on all cells, if you have a CAR T that's targeting it, it's going to kill all the good cells as well as the bad cells. Also, it's going to kill themselves. The T cells express CD45. So the T cell targeting CD45 are going to commit what's called fratricide. All right, how do we get around this? Work from Carl June, who is one of the OGs in this approach, going to win the Nobel Prize for sure, along with Sar Gill, both at UPenn. They built on this prior work, all right? And this is in treatment of AML, right? So AML is targeting CD33, but CD33 is present on CAR T cells, right? So how do you treat the tumor without having this fratricide? What they did in that case is that they knocked out CD33 in the CAR T cells, so there wasn't the fratricide element. Okay, could you do that with CD45? No, right? Because if you knock out CD45, you get impaired T cell development and function. And also, by the way, severe combined immunodeficiency, also known as SCID. You don't want that, right? So they had to come up with a nuanced approach. And I thought this was really brilliant, conceptually. What they did here is, instead of deleting CD45 altogether, they used CRISPR base editing to install or to modulate the CD45 epitope, all right? So they put in this mutation that changed the antigenicity pretty much of CD45, right? So CD45 was still functional, but it didn't look like the native CD45. So the CAR T cells didn't attack it. They call this approach epitope editing. And they used it with great success. They showed that these epitope edited CAR T cells, they didn't kill each other, no fratricide there. And they were also effective in treating AML, B-cell lymphoma, T-cell leukemia, so they could attack pretty much all the different hematological malignant cells that are out there, or multiples that they tested here. And critically here, the native or the epitope edited hematopoietic stem cells were not attacked by the CAR T cells. So you could engraft those cells, they would persist and differentiate in vivo into the whole complement of hematopoietic derivatives, but the CAR T cells wouldn't attack them. So the idea here is you're effectively changing your entire hematopoietic complement, or introducing a new hematopoietic complement while also introducing the CAR T cells that are going to wipe out every other blood cell in your body, including the malignant cells. I think that's a really brilliant conceptual advance and could create a more off-the-shelf, I guess, product, which will be patient-specific in every case, but it won't have to be tailored to each specific malignancy. It can just treat blood as a whole. So I think that not just in the blood, but maybe this approach could be applied toward other organs, although not as easily, because you can't exactly replace your native organ complement with epitope editing. But I thought a neat conceptual advance and a nice hack following from your hack there, the fetal globin. What do you think?
Yeah, this is a really interesting idea. I think this idea of epitope editing is a really unique gene editing strategy. So you can protect cell therapies from these, like, from targeting themselves and all these other applications. But they mentioned in the discussion, there's potentially other applications, and you could use the platform for like in vivo selection of genome edited HSCs without genotoxin conditioning, or maybe even that they allude to this eradication of the latent HIV reservoir. So that's a really powerful application. But and then they're thinking about other applications of this epitope editing to like other targets that we've actually talked about recently, CD117 or maybe CD123. So really cool technology here. And again, I've mentioned it before. I feel like we've become an offshoot of the immunology podcast here. We're covering so many of these stories now related to, hematopoietic stem cells, editing of these things, that maybe we should just merge into one grand podcast, just the podcast, right? But hey, it's just a reflection of how, important these two fields are and how well intersected these two fields are, especially in the clinical applications too, right? The story I previously talked about was a clinical trial of HSCs and edited HSCs, and inevitably this is going to move towards clinical trials as well, this epitope editing approach. I also think we need to have David Liu on the show at some point, and we talk about all of these prime editing based approaches that they're working on over there in Boston. So Dr. Liu, if you're listening to this, we'd love to have you on.
Yes, we would. A lot to talk about as all these editing technologies become so pervasive. But I just will remind you, Arun, as someone who loves the blood and also loves the immunology podcast, that it is the original stem cell and the first stem cell therapy. So I think there's plenty of stories for us to share. And I would love a merger for one of these shows. We could make it like a 5-hour bonanza of everything stem cells and immunology. Can't wait for that. I'm gonna have to slate that one for episode 251, perhaps.
Let's see. Maybe let's, back off from the merger talks. We'll stay in our own realms. But hey, I love crossover episodes. So, Brenda and Jason, if you're listening, love to have you on at some point, in addition to Dr. Liu, of course. Moving on to the last paper of the roundup for today. This is not related to HSCs for once. This is a reprogramming story, a basic reprogramming story, but I think has a lot of really interesting implications here. This is I'm actually sort of shocked here. I didn't realize that reprogramming stories 10 years later, 15 years after IPS reprogramming was first described, I didn't realize they would still have enough in them to actually merit a nature study to come out of it, right? But hey, this is super important, and I'm not belittling this at all. I was just a little surprised that even reprogramming in this day and age can merit this level of publicity and this level of caliber of study. But it also is a reflection of just how important and the rigor of this approach that they took here in this particular story. This is coming out of the labs of Jose Polo and Ryan Lister, right? First author here is Sam Buckberry. This is a study looking at reprogramming and how it could be improved even further to the level of having iPSCs truly on par with human embryonic stem cells. Okay. And we're talking about epigenomics specifically. iPSCs, I think, we've run into this fallacy in the modern stem cell age that iPSCs and ESCs are equivalent. And for all intents and purposes, for the downstream, they are largely equivalent, but epigenetically, they're still quite different. Okay. So we know that cells undergo this huge epigenome reconfiguration when they're reprogrammed to iPSCs from somatic cells. It's just like the saying is they go back in time, right? That's a simplification of the fact. But iPSC and embryonic stem cell epigenomes are still very, very different, even if their downstream production of, say, cardiomyocytes, whatever, is comparable and their efficiency of differentiation is comparable. But epigenomically, these cells are still really, really different. So first, what they did here, first they did a just really deep characterization of the epigenetic differences between iPSCs and embryonic stem cells, human iPSCs and human embryonic stem cells, did a bunch of genome-wide DNA demethylation or methylation profiling through the primed and naive reprogramming of human somatic cells to iPSCs. And they found that there are reprogramming-induced epigenetic aberrations that emerge midway through primed reprogramming, which is pretty much what almost everybody out there does, is primed reprogramming. But DNA methylation begins early in naive reprogramming. So using that knowledge and using that amazing basic science skill set, data set, they developed what they call TNT, really a bombshell here, pun intended, I guess, right? Transient naive treatment, or TNT-based reprogramming strategy that, ultimately it's hoping to emulate the embryonic epigenetic reset, ultimately making iPSCs that are very similar epigenetically to embryonic stem cells. Okay. And the thing I really liked about this, and I didn't read super deep into the paper, but I just wanted to emphasize this fact. What they did to actually create iPSCs that are on par epigenetically with human embryonic stem cells is just modulate the media in which they were placed during the reprogramming process. So they had a naive state media and they had a prime state media. And just by modulating the amount of time the cells were in the media, that's how you got these epigenetically superior iPSCs on par with the embryonic stem cells. Okay. And then they demonstrated that these TNT iPSCs can, differentiate very similarly to the human embryonic stem cells, that the differentiation efficiencies are similar. And it can actually, in some situations, improve the differentiation of iPSCs derived from different cell types. And so their ultimate conclusion as this TNT reprogramming corrects epigenetic memory and certain aberrations like transposable element overexpression, that sort of stuff that's present in iPS reprogramming traditionally. So this TNT reprogramming makes these cells epigenetically superior, that are what they say molecularly and functionally more similar to human embryonic stem cells than conventional iPSCs that we're using every day in our lab. And then they end, by looking to the future and thinking that, if TNT reprogramming is really making these improved iPSCs that are epigenetically on par with ESCs, then perhaps there's a new standard now. There's a new standard for using these iPSCs for downstream biomedical and therapeutic applications, maybe for clinical trials like what we alluded to earlier, or these, TNT iPSC derived cell products that maybe Blue Rock could use in the future. Who knows? Maybe they'll be even better at making the dopaminergic neurons or whatever cell types that you want to use for clinical applications. So, I just wanted to reemphasize again, I'm not belittling this study at all. I was just surprised that we have, enough that we're learning about the programming process, even now in 2023, to have a paper in the caliber of a Nature paper. But there's just so much in here, and I think the implications here are so important that it's definitely worthy of this level of publication. So kudos to the Lister and Polo groups for this amazing work.
Yes, explosive is another word for it.
Yeah.
I, like you, when I started reading this, was like, oh, I can't believe there's still anything left in the tank. with these reprogramming groups. But yeah, then, as I read it, I was impressed with, as you reviewed there, the power of just observing the system and then having this relatively, I guess, minor intervention that's, no barrier to entry for labs out there, so it can be widely adopted. I guess that's how it gets to this caliber of a story. It's something that is simple, but has, I'd say, outsized ramifications. And considering our guest today and how we're moving into this kind of embryo modeling space, where to date no one has been able to create a blastoid or other type embryo model that could actually generate a live born and or fertile offspring. I think there is still some things to learn. I know the gold standard is the tetraploid complementation, and that's not in human, but in mouse it's been very well established that these cells have the capacity to undergo tetraploid complementation, demonstrating their true pluripotency. But there are still some open questions regarding whether or not those cells faithfully mirror their physiological correlate from an epigenomic standpoint, and that may be relevant to disease modeling or whatever else in the future. So I think it's important that this study was done. I think it's great that there's now a really easy recipe out there for people to kick around and test whether or not this new explosive recipe can actually engender any kind of phenotypic differences or capabilities amongst the ES-derived products. So I think it's a great story. I never thought I'd read another reprogramming story again in Nature, but here we are.
Yep, here we are in 2023. TNT is the wave of the future, absolutely. I mean, one thing that I thought was, potentially interesting here, and they didn't really characterize this differentiation in this paper. They looked at a bunch of cell types, from these TNT iPSCs, like the cardiac and the endoderm differentiation approaches. As we know, and as you know, the epigenome is so critically important for the production of bona fide gametes, right? Sperm and egg, right? And that's certainly been a huge limitation in making bona fide gametes from human-induced pluripotent stem cells, is perhaps that epigenetically, whatever, right now the cells we're using as the cell population to make those cells, it's just not right? And so maybe these TNT iPSCs are the key to making those bona fide germ cells from human iPS cells. So we'll see what these folks do next with this really explosive technology, right? Pun intended, I suppose.
Yes, I'm gonna really bury the pun here by saying, I've been outspoken that we should not be making gametes from pluripotent cells. You're getting me to get out on the limb again, and thanks for that. But I certainly wouldn't start with a recipe that has TNT in it, my friend. That could have an explosive outcome. But enough of the puns. We're going to get to our conversation with Dr. Nicolas Rivron in just a minute. Before we get there, I've got a quick message from STEMCELL Technologies. Looking for more information on organoids? Download STEMCELL Technologies' new ebook on organoid research techniques, developed in collaboration with Wiley Publishing. This essential knowledge briefing details the evolution of organoid technologies from discovery to application, including discussion of key milestones and advances of the technology. A review of key publications and annotated reading lists provide further background on many of the topics covered. Download your copy at www.stemcell.com/organoid-ebook. All right, guys, today we have a terrific guest for you from the IMBA, that's the Institute of Molecular Biotechnology of the Austrian Academy of Sciences in Vienna. We have Principal Investigator Dr. Nicolas Rivron. I really don't even need to introduce him. He's a star in the field, really single-handedly, well, not single-handedly, with his group, along with other many esteemed researchers, brought the embryo model into the fore. The Rivron group recreates embryonic development using mammalian stem cells in a dish to better understand the encoded principles of self-organization. And we're going to get into all that with him in just a second. Nicolas, thank you again so much for joining us here on the show.
Hi, everyone. It's very happy to be here.
Yeah, welcome back. And I mean, let's just jump right into it. It's no secret that the modeling of embryo development in vitro has emerged as, in my mind, the hottest subfield in stem cell biology right now. I mean, I've gone so far as to say that if I was a trainee all over again in the field of stem cell biology, I would be doing this. This is what I would be working on. So, I mean, it's very prominent, and it's in large part thanks to these amazing new technologies, such as the human blastoids that your lab has developed. And the amazing thing about these technologies is that they're reproducible and scalable. So your Nature paper on using human blastoids to actually model blastocyst development came out about 2 years ago. But why don't we start off actually just by recapping what your lab is working on right now to make some of these in vitro models even better?
Yeah, so we know we started a few years ago to try to reform those blastocyst-like structures, and we started with the mouse one, and because it was a complete model, we could transfer it back into the mice, and it was quite an exciting experiment, although it totally failed because we were never able to form a mouse. But it started something that where we started to think about what are the minimal principles that are necessary for an embryo to actually develop, and we can use this approach in order to better understand those principles that are so hard to grasp. And so that was in a couple of years ago, and then we moved to Austria. And there we tried to reproduce the same concept using human cells, and it does not happen often in life, but it turned out to be relatively easy, in the sense that it happened quite fast, and it happened with very high efficiency. And here we are actually standing on the shoulders of many other labs, because there was so much work that was done before to capture those so-called naive human pluripotent stem cells. And we use those cells, and here this is the work of 3-4 people, in fact. The first one is Harunobu Kagawa, second one is Alok Javali, third one is Heidar Ikoi, and the fourth one is a trainee at that time that was doing her master studies, which is Teresa Sommer, and who's now still in the lab. And those 4 people together, they just pulled it off very rapidly, and it was very surprising, because I still remember the first time Alok Javali came back to us saying, "Look, guys, I think there's something in the dish here." And we were like, okay, no, it's not happening. Just do a triple staining of those 3 cell types that are known to be inside the blastocyst, and we'll check it out. And he came back 2 days later with the 3 cell types that popped up in those blastoids. And so that was a very exciting moment. So this is for building the models. And now the question is more we are. I think the field is a little bit beyond building those models. There's still a lot that is being built, a lot, and this is very important that we have a whole variety of models that are built, that are complementary and more or less complete, and representing different stages. But we are slowly drifting into actually doing good science with those models. Yeah. And trying to answer proper questions that could not be answered until now. And there are many questions that can be answered, but I think in our case, what's very exciting is that we can model the moment of implantation into the uterus, and because those blastocyst-like structures have they are complete and have the outer part. They can actually be transferred into the uterus in the case of a mouse, and we can play with the model in order to see what's necessary for it to develop. And in the case of the human, they can be put in interaction with uterine cells in a dish. And we did this in 2021, and it was very surprising. That was probably like the second wow moment in the lab, because Heydar Ikoy, he did his PhD on endometrium organoids, which is the lining of the uterus, and he started developing those implantation assays, and he realized very quickly that actually the blastoids they could very specifically interact with endometrial cells, and they would only do this if they were stimulated before with the hormones of pregnancy, progesterone, estradiol, and also a Wnt inhibitor that Margarita Turco had found out before, that primes the cells for implantation. And so when Heydar was depositing those blastoids onto those layers, nothing was happening, except if you were stimulating them with hormones and this Wnt inhibitor. In that case, you had about 40% of the blastoids that attach. And then Heydar went to the microscope and did some live imaging. And what you observed is that the blastoids, they not only attach, but they also roll around, and they find their way, and they only attach via one side. It's always the same side. And of course, this is known to be the sticky side of the human blastocyst. So, there was a very strong specificity, not only in the endometrial cells that had to be hormonally stimulated, but also with the blastoid that had formed spontaneously an axis and formed those sticky cells. And so that was, this is when we realized that we can really start tackling really interesting scientific questions. And there are a couple of sorry, I'm a bit long with that, but there are a couple of controls that we made on the side that were quite interesting, because we really need to understand what those models are really doing, and if those models are really recapitulating biology. And so when we saw that the human blastoids were attaching via only one side, it led directly to the hypothesis that the inner part of the embryo, the future fetus, is actually signaling locally to the outer part of the uterus, which is the trophoblast, to make them mature and make them sticky. And one very good experiment that we pulled off on, I thought was really cool, is that we could inhibit the formation of those inner cells by using a hippo overactivator. It's called XMU MP01, or a STAT inhibitor called SC144. And in both cases, if we put this single molecule during blastoid formation, we actually don't form a blastoid, we form an anti-blastoid, a trophosphere. And when we deposited those trophospheres onto the hormonally stimulated endometrial cells, they were incapable of attaching. So it really fits. It really fits with the idea that at the very beginning of life, it's actually the inner part, the future fetus, the future embryo, that is instructing the outer part. That is the future placenta, in fact, to become sticky locally and to implant into a specific direction into the uterus. So this is a very interesting process in the mammalian embryo as a design that evolved in order to form those outer cells that are going to mediate the implantation into the uterus and later form the placenta. And by design, it is pushing those cells to develop, and we found many factors that promotes this. And it is locally directing those cells to become sticky, to implant in the right direction into the uterus. So these are all kind of interesting things that we can now study in addition. And one quite interesting aspect is that, in fact, the mouse blastocyst is implanting via the opposite direction as compared to the human one. So, those sticky cells that I was telling you about, they actually form on the opposite side in the mouse blastocyst as compared to the human one. So somehow during evolution, the whole system has flipped around by about 180 degrees. And I still cannot put my head around this. How is it possible that, somehow those embryos flipped around and still managed to develop and formed like this new way of developing inside the uterus? This is one of like those jumps in evolution that for me is like completely blowing my mind. So these are the kind of things that we can now like precisely like decipher in a dish, and then try to like look at all the signals, all the gene regulatory network, all the the principles and the evolution of this from species to species. This is really cool,
Yeah, I mean, cool is an understatement, and that to me is what's so exciting. What you just highlighted, all that, as someone who focuses on reproductive biology, is that the diversity of strategies for reproductive success across mammals, it's very broad. Even with closely related species, it's not like the heart where the pump is effectively the same across the board. Like you just described there, you can have these vastly different, when the embryo implants, what the poles are of the embryo during implantation. And these are all things that we never could really address without getting into that black box of the uterus. So that's one innovation that I think was broke open with these blastoids. But also, as you just alluded to there, with the difference between mouse and human, this is stuff that we can't even model these phenomena in the animals that we're accustomed to, or we can't model them in a way that actually recapitulates the human biology. So I think that's what's so amazing about this, is not the making an embryo per se, but trying to unpack the process of implantation using these as a model. And along those lines, I guess I don't want to say disappointed, but it's frustrating for me, I guess, that a lot of the reaction is so focused on making, how late can these embryos go? Can we make organs? Is this going to be making humans in a Brave New World type scenario incubator? It's like a lot of the hype and maybe scandal or skepticism, whatever you want to call it, that surrounds the work, I think is really missing the point, that this is an amazing scientific tool. But I gotta ask, I mean, you had alluded to it in your intro there, that you could implant these in the mouse and get implantation. I think a lot of people out there waiting to see live and maybe fertile offspring derived from mouse embryo models implanted in foster mouse uterus. Maybe these mice are even already alive and in review in some journal somewhere. But the published data suggests that as ordered as the embryo models look at peri-implantation stages, something goes off track. Do you have any ideas as to what the problem is there and how we can get over that obstacle to actually get live born pups from these embryo models?
Yeah, well, I hope the mice are running somewhere. And I'm sure there are plenty. I hope there are plenty of people trying. This is not really like a big focus of our lab. We are more interested into like the questions related to like implantation, but clearly we still need to understand what are the limitations of the model. So we are clearly like working on those questions of why is it that it does not develop to a certain stage, at least in utero. And the first thing that we realized is that the trophoblast stem cells that we were using, which have been derived by Janet Ronson, and like that was in 1998, are actually like beautiful cell lines, but they are very heterogeneous. And they comprise some subpopulations that are blastocyst-like, but also some subpopulation that are post-implantation like. And there's actually like 10% of the cells that are already like very differentiated. They are almost, like at the verge of like, post-mitotic stage. So one thing that we did in the last few years, and we published the paper in 2022. This is the work of Jin Woo-seong in the lab, along with a PhD student who is called Vicky Holzman. And together, what they found is that you can also capture trophoblast stem cells in a more pristine stage that better reflects the blastocyst trophectoderm. And we did this by learning from the embryo, in fact, because what we observed initially is that when we form blastoids from trophoblast stem cells, the trophoblast become normalized over time, meaning that they become more blastocyst-like over time. And we speculated that this is because they are in the presence of the embryonic cells that must be sending some signals. And so we actually looked at all the signals that are coming from the embryonic cells, and then we run some combinatorial screens on trophoblast stem cells using those molecules that come from the epiblast. And we found one combination that allows to capture what we call trophectoderm stem cells. And those trophectoderm stem cells much better reflect the transcriptome of the blastocyst trophectoderm, and they can form better blastoids also. And what was interesting is that when we placed those blastoids, those new blastoids within the uterus of mice, they started to implant at much higher levels. In the original publication, 2018, it was 8%, meaning that 8% of the blastoids were actually instructing the uterus in a proper way so that the uterus reacts by forming the cocoon around it, which is called the decidua. And with those new version of blastoids made with trophectoderm stem cells, it goes up to 20-25%. When you do the same procedures with blastocyst, the real thing, you are at 40%. Because of the surgical procedure, not all of them are implanting. So we go from 8 to 20, closer to 40. And of course, it becomes much easier for us because we can get a lot more of those deciduas. So practically, it's so much easier to analyze things, and we can generate samples. And the second thing is that we started to look at the difference between those blastoid 1.0 and 2.0. And what we observed is that in those trophectoderm stem cells, they were upregulating a key homeodomain gene that is called CDX2. And that when we analyze the chip sequencing data, we saw that CDX2 is very strongly binding to Win6 and Win7b. And when we knocked out those genes in the blastoid, those blastoids dramatically decrease their efficiency of implanting and instructing the uterus. So, and we did a couple of other experiments around this, but all this is pointing at the fact that in order to implant into the uterus, the blastocyst is producing Win6, Win7b that is playing a major role in instructing the uterus to decidualize. And very, very interestingly, those molecules are also produced by the human blastocyst. So it's possible that there's a conserved process here by which, despite the opposite mode of implantation, there are some commonalities, such as expression of Wnt into decidualization. So this is one thing that helped us to bring blastoids to much higher efficiency of implantation. And the second thing is the primitive endoderm. So there's a second extramyelic tissue within the blastocyst, which is called primitive endoderm, and that is going to form the yolk sac. And in the original blastoid, there was a primitive endoderm. There were actually the three lineages of the blastocyst: trophectoderm, epiblast, and primitive endoderm. But the number of primitive endoderm was very few, definitely less than what is normal. And if you don't have a primitive endoderm, we know from classical embryology that nothing will develop, because this primitive endoderm is encasing the epiblast and is promoting its development. So there was we actually found new ways to boost the formation of primitive endoderm, and we published this in 2022 also, I think. And we can to better extent have development of the epiblast. But what we are very interested in is to understand what is the crosstalk now. What makes this synergy between those extramyelic tissues and the epiblast. Yeah. So overall, we learn by doing, and we find all kinds of defects in the original blastoid that we fix step by step, and hopefully somebody has fixed all those defects already, and have some mice running somewhere in a lab, and yeah, looking forward to read that paper.
Yeah, I think we all are. I mean, the fact that you're able to actually progress the development of these embryo models just by implanting them in vivo, that's just so fascinating to me, and just a million questions that are unlocked from that capability. It's just, as somebody just who's outside of the field, I'm just blown away that this is actually possible. So I'm just so excited to listen to that. I guess shifting away from the science for a little bit, I wanted to talk about a recent development in the field that's non-science related. I guess it's tangential. Of course, we had the ISSCR annual meeting not too long ago. You're of course involved with the ISSCR in drafting some of the guidelines for early embryo models, for example. And it was just a case point in how prevalent this subfield of stem cell biology has become, is reflected in the very first plenary talk of the conference from Magdalena Zernicka-Goetz, who of course you know pretty well, where she presented some of her recent work. And we kind of know what happened next. After that presentation, there was a number of other preprints that got released online during the course of the conference. And it's just such a huge flow and influx of data that's coming out from these studies. And oftentimes they're happening all at the same time. Things are getting pretty competitive, to be honest with you. I mean, we had Jacob Hanna's group, who also published similar work and, had their preprint go up as well. I mean, things are very fast moving and very competitive in some situations. What are your thoughts on the pace of the field, of how quickly and rapidly it's progressing?
I think it's important that there's a lot of groups that are involved, and because by having multiple groups, then we can progress better, and we can actually form different types of models, but also try to understand what the models are forming, and we need to set up very good standards in the field. We need to make sure that we are producing the right cells at the right moment, and that those cells pattern and form the right tissues in the correct way. So there's a lot of, it's super important that we actually are able to really understand the structures and the cells that are formed. So for this, there's a tool that's super powerful. It's a single cell transcriptomic, of course, and this became a standard for everybody to do a single cell RNA-seq to the minimum. And but there's very few references that we can benchmark our models to. So there's like a very important need for developing like good reference maps of human embryonic development at the single cell level. This is going to be absolutely crucial to understand, what the models are forming and to which extent they can be useful to do science. But beyond the single cell transcriptomic, there's also like a lot of need for doing better quantitative morphometric studies to understand, at the single cell level, how similar the cells that are formed and the tissue that are formed are to the real thing. Of course, for humans, for human embryos, it's extremely complicated there. But once we have designed proper ways for human embryos to develop in the dish, those IVF embryos that are generously donated by those couples that have finished their parental project, we can try to like grow those human blastocysts in a dish, and for them to form the right cells at the right time and the right tissues in the right places, and then get some very good morphometric data from those in vitro grown blastocysts, and to do some high content imaging, and we can obtain like very large data from this high content imaging, literally like hundreds of parameters per cell, and draw again those maps in order to like nicely understand what's happening here. So this is something that I think as a field that like we really need to like establish in the next couple of years, and there are already like quite some people who are making efforts into doing this, and we need to reinforce this. Like the models that they are only going to be, they are going to be useful if they recapitulate, if we know what they recapitulate. So it's not that they have to recapitulate everything, but we have to know what they recapitulate. And once we know, this allows to focus the science, and to like answer like those questions that we are after. So like it's work in progress.
Yeah. I can only imagine the kind of progress you got going on in your lab right now, but I want to get down to something, specifically clinical. I know your interest is in self-organization, just the phenomenon of self-organization in systems. And you're focused on this peri-implantation stage and the cellular interactions. And you elaborated there on the education, so to speak, that the ICM gives to both the trophectoderm and the interaction there with the primitive endoderm. These are all cells that are present in vitro fertilization, right? And this culture period that the embryos undergo before transfer back into the uterus. Are you investigating in that you have this model and you've already gleaned some insight into the signaling between? How do you apply that clinically to in vitro culture of human embryos? I mean, can you? I feel like you could do that right now. Do you have any kind of capacity or your research lab that's just really focused on that translational approach?
Yeah, totally. Like so like every year in the lab we have like the carrier meeting, like in many labs, and in 2021, Alok Javali, who was one of the co-first authors on the 2021 paper, he came into my office, and he was like, like, I'm not completely sure if I can actually like continue to do science for the rest of my life, because at that time what his reasoning was that he's Indian and he wanted to go back to India at some point, and he thought it's not going to be like super feasible for me to actually like go back to India and do stem cell research because it requires a lot of money. And so, I'm actually thinking about like doing like something more translational and more entrepreneurial. And I was like, yeah, well, why don't you try? And we actually like spent quite some time since then. And he has set up a company that is still in the making, but that I think is doing very well, and that is using Blastoid technology along with implantation assays, as we developed previously, in order to tackle clinical problems. And Alok is actually the managing director of the company, and he's running the show over there. He's absolutely amazing, that guy. And so we are having a very good time because we've discussed tremendously with clinicians in the field in order to like understand what can be done and how we can help. And we came up with like a couple of clear problems and started screening. And so we are actually like in the process of testing a couple of things that came out from Blastoid research in the preclinical setting at the moment. So he has like teamed up with a couple of clinicians, and he's actually like already like working on seeing how predictive human blastoids are of human blastocysts. I think we're going to get some surprises here, because so we can easily find a lot of principles. We can easily find a lot of molecules, but like going through the whole process of actually like getting to contribute to the clinical world is a long way. But yeah, if there's one person who can do it, is definitely Alok Javeli. So I think you have to like you have to know why you're actually doing this, because it's a tremendous effort to set up those more translational aspects. In our case, it's because I really want to know if blastoids are good enough to contribute to clinical research. And for me, like proving that blastoids, the science that comes out from blastoid research is good enough to contribute to clinical research would be a big plus. It would mean that somehow the models are good. So like we don't know where this is going, but it's a lot of fun, and Alok is pretty geared up, so yeah, we'll see.
Yeah, I guess, again, a lot of fun is an understatement. Just as a third party listening to you talk about this stuff, it gets me excited, and I think it's actually a good time now to talk about some of the guidelines, right? So you've been involved in developing some of the ISSCR guidelines and recommendations concerning research on human embryo models formed from stem cells, and actually had this Stem Cell Reports article on the topic a few years ago. I mean, as you just alluded to, for example, your colleague from India, the stem cell research community is an international one, and researchers from the different parts of the world are subject to different laws and different regulations, especially when it comes to this specific type of work. So tell us why it's really important to have guidance on what should be considered appropriate or not when it comes to this sort of early embryo modeling work. And also, maybe what's the next step when it comes to the evolution of these guidelines?
Yeah, no, this is more my part, like this, and I put a lot of thoughts into this, because I think it's super important, and it's also very interesting because it brings you to a different space. We just talked about the clinical space, but those ethical aspects, it brought me to discuss with ethicists, with philosophers, with people in the regulatory space. And this really opens up your mind and your views on things. That has been like extremely interesting. And the first thing is that, you know, like, so in 2018, when we published the mouse blastoids, we very rapidly gathered a couple of people in order to think about those issues. And here I have to give credit to a couple of more senior people in the field who really like put some effort into making us think about this. And I'm talking about Martin Pera. I'm talking about Janet Ronson. I'm talking also about Amanda Clark. And they actually like gathered a bunch of people within the ISSCR to think about those questions. And this lasted for 2 years. For 2 years, we had like dozens of discussions with people all around the world. It was about like 15 people. And we thought, okay, we need to like come up with like an update of the guidelines that tackles the urgent questions. And one of them, and the reason I was invited, it was that we knew that human blastoids would be on the way. Of course, they were on the way in my lab, and they were on the way in multiple other labs. And we wanted the ethics to be done in parallel of the science. And it turned out to be like very successful, because in 2021, the year where the human blastoids were published, the guidelines also came out. And in those guidelines, there were a couple of things that we said. The first thing is that this research should have an ethical oversight, and especially the research of what we called integrated embryo models, which are models that comprise the two extramedullary tissues, and that might develop, when improved, might possibly develop into a fetus, the model that will develop into the fetus. And this type of research should be like, you know, like supervised by the same type of committees that is used for human embryos. On the contrary, there's non-integrated models, and those ones, there are less ethical concerns, and they can be supervised by the committees, but don't need to have an authorization to do this here. And the second thing that we mentioned is that we should prohibit that human embryo models are transferred into any uterus, whether this is animal or human. And those rules that are ISSCR guidelines actually came out, you know, right on time. And it was super important that, you know, those things are done in parallel. And there are a couple of other additional things that we put, but these were probably like the most important ones. And since then, to answer your question, we also kept on thinking about what are the next things to do. And we have like a paper that should come out in the next few weeks about this, and that tackles a couple of important questions for us. The first one is to understand why do we consider that embryo models are not embryos. It seems quite trivial for scientists, but, you know, like thinking a bit more deeply about it is showing that we must refine the definition of the human embryo. And we came up with like a couple of thoughts of how should we define those human embryos. And you know, we know for a long time already that embryos can be formed not only by fertilization, but also by other means. They can be formed by somatic cell nuclear transfer. They can also be formed by transferring a set of chromosomes into an embryo, at least in the mouse. they can also be formed by forming gametes in vitro from stem cells and fertilizing those gametes in a dish. And now we know that they can also potentially be formed by just self-organizing stem cells. So, like the question is when. So how should we consider all those different type of embryos? And what my thought is that we should consider them as embryos as long as they are capable of forming a fetus. And that if this is proven, then automatically the structure tips over and becomes fully entitled to be, like legally quite similar to a human embryo. So, the question is when would those embryo models eventually tip over to become embryos? This is not, we are not close to it, but I think we should start thinking about this. And of course, because we cannot transfer embryo models into a uterus, we cannot do the ultimate test. So we have to find ways around this. And what we'd like to propose is some Turing test, by the name of Alan Turing, who was like designing those tests for computers and artificial intelligence. Like it was like thinking of, okay, like if a blinded tester can ask questions to a machine and be fooled by the machine by thinking that he's actually talking to a human being, then the machine has a level of intelligence that is sufficient to, to give it credit. And we are trying to think of like Turing test for embryo models that by which we could biologically test those embryo models in order to see if they pass those tests. And so there are like two tests that we have in mind. And the first one would be, of course, if those models could develop efficiently and faithfully in a dish until a certain point. And if this is happening, then you have a certain level of certainty about the quality of your model. And here the question is, when do we actually stop the in vitro experiment? And this would probably have to be a compromise between, the longer you wait, the more sure you are about your model, but you don't want to wait too long so that you run into ethical problems. So that's probably the first Turing test. But I don't think this is sufficient for embryo models to tip over, because you really want to know if they are really going far enough to form a fetus and eventually to have the capacity to form a neonate. So I think next to this, there's a second Turing test that we can think of, which would be if a similar embryo model in an animal would be able to form living fertile animals. And here, of course, we can start with mice, but mice are very different from humans. So I think there's a necessity to put the bar a little bit higher and to think of recapitulating full embryogenesis with species that are closer to humans, such as pigs and non-human primates. And if an embryo model is capable of forming living fertile pigs, living fertile non-human primates, then the second Turing test, in my view, is passed. And if the two Turing tests are passed for a specific embryo model, then I would tend to think that this specific embryo model should probably tip over and be legally considered similar, if not equal to a human embryo. And after, there can be multiple additional layers to be added to this. It's not because you are legally considered an embryo that you can do everything with such an embryo. For example, the somatic cell nuclear transfer embryos are considered embryos, but they cannot be used for reproduction. But they can be used for research in some countries. Yeah. So we would have to then think about what we actually do with those embryo models, potentially turned embryos, and establish additional layers of regulation in order to know what is acceptable and what is not. And I think, for example, it's clear to everybody that it's not acceptable to transfer those embryo models into a uterus because it's probably going to develop abnormally, and it could harm the gestional carrier, such as like in the case of molar pregnancy or ectopic pregnancy. But it could be that they are still allowed for research because these are important models, and even if they are legally considered similar to an embryo, we still consider that they should still be used for finding drugs, improving IVF or such things. So these are the things that we are currently thinking of. Like what is the definition of an embryo? Can we actually refine it based on in the light of embryo models? Once we have this refined definition, can we use it in order to define precise tipping points for when eventually embryo models would become embryos, legally speaking? And what are the Turing tests for doing so? And then finally, what are the different additional regulations that we want to put in place to ensure that the use of those embryo models is, following ethical guidelines and carving an ethical path that would maximize benefits to society. So these are the things that we are thinking of now.
Yeah, you've got a very narrow scope, right? Not thinking big at all. But the impressive thing to me, I mean, everyone gets so caught up in the humanness or the embryo-ness properly, as you said. This Turing test for the embryos are important. I think a lot of experiments to come are going to flesh that out. But whether or not these things can create a fertile fertile being in whatever species, the maxim holds, George Bach, all models are wrong, some are useful. And I think that's the key innovation here with your Blastoid and all the embryo models that have followed, is that you kicked open a door that now we can ask all these questions. All these things you've talked about are things that were totally inscrutable, impenetrable. And now we've got experiments that we're designing to get there. So I think the progress has been tremendous, and it's very exciting. But what's more important to me as someone who's studied embryology my whole adult life is that we're finally seeing these things firsthand, and are able to exert the power of observation, experimentation on them. So very impressed and appreciative of your seminal contribution, Nicolas. And before we let you go here, we're going to ask you a couple peripheral questions. The first one is, what's one piece of advice you've been given, professional or not, that you would like to pass along?
Yeah. Yeah, I didn't really think too much about it, I have to say, but so yeah, we can maybe doing it on the fly is a good way to get the things that is on that I think is important. I think there's there's a lot right now. There's a lot of hype in this field, and there's a lot of competition also, but like first, we have to make sure that we do things right, and we have to make sure that we do things well, and we have to make sure that like the people that are working on those embryo models in the labs are actually like enjoying it and really like are being driven by the science, like the hardcore science, the will to do discoveries, like so I'll give one quote. I'll give one quote from Janet Rosson, who is like like always very insightful, and like I remember her like telling me like like blastoids can be nice if they form a mouse, but most importantly like you have to fill the gaps in knowledge. You have to like use this system in order to like fill the little gaps that needs to be filled in embryology. It's kind of like there's like, if you like represent like knowledge in the form of like a three-dimensional tree with like the core is like what we really know. We know, and we are standing on this knowledge, and there's no way that we can actually like challenge this, and I think this is very important to remember, that there is this core knowledge, and that's like we are standing on this, and this is firm. But then after, there's like the branches that are like like becoming more and more spaced. And this is what I think she meant by filling knowledge gaps. It's not necessarily like the like the most edgy questions in science, but it's super important that we actually like understand this like very finely, and that we feel those gaps, and this is what I was talking about, Win6, Win7b, like, well, this is classical signaling. This is not, there's nothing like super edgy about this, but this is really important. We need to like use this in order to like understand the basics here. And then after, if you look at the very external part of this three-dimensional tree of knowledge, then there's the thing that is really at the edge here. And here there's quite a few things that can be answered, and like we are certainly like tackling. We think we are tackling some of those questions, but like finding a good question is very complicated, and like it takes a lot of years. So my advice would be just like be happy like filling the knowledge gaps, just like Janet said. And over the years, you're going to find those cutting edge questions that are at the tip of knowledge here, and then it's going to become even more fun. So yeah, just enjoy the process.
Any advice from Janet Rosson, I think, is worth considering. That's for sure. And finally, if you weren't doing what you do in the lab as a scientist, what do you think you'd be doing with your life?
Oh, yeah. Yeah, I'm thinking a lot about my retirement, So, so like, no, I'm very happy doing science. I'm very happy doing science, but I'm also very happy like changing fields. And so this is probably like, I would consider this as a different job. So it would still be in science, but I'm very happy to like venture from one place to another, because you learn so much from this. So I was trained as an engineer. I did my PhD in the field of tissue engineering, which is very far from what I'm doing now. Then we ventured into embryology and stem cells, and now I'm becoming very interested in evolution, and into like like what are the main drivers of this divergence between species, and how this might have impacted the different ways that embryos formed, and what makes human embryology so frail as compared to other species. Why is it so that like humans are so suboptimal in doing so? So I wouldn't call this a different job, but I would certainly be very excited about like venturing into this type of space. Sorry for not answering the question.
No, that's an answer. Third act in Evo Devo. I can't wait to see. I mean, you've already blown our collective minds with your science. I can't wait to see what comes of your theory, Nicolas. But I guess I'll have to wait a bit longer because you still got some work to do with them blastoids and embryoid models, my friend. And we can't wait to have you back on the show soon enough to tell us how that pans out. But for now, we're finished, and very grateful for sharing, just your thoughts, your journey, your process, and all of it. Really appreciate you joining us today.
Thanks so much, and thanks for doing this. It's awesome.
All right, you guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all of the interview and roundup papers. You can also reach out to us on Twitter at StemCellPodcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. This has been a great episode. Until the next time, thank you so much for listening.